Earlier we mentioned that speciation usually requires some form of a reproductive barrier, something that separates the two groups to where they begin to diverge from one another. Oftentimes this is a geographic separation, but it doesn't have to be. So we're going to look at those concepts. Concept 24.2 says speciation can take place with or without geographic separation. And there are specific words for these ideas, so we're going to look at those. Speciation can occur in two ways. Allopatric speciation. The word allopatric is from the Greek allos, which means other, and patra, which means homeland. So allopatric is usually thought of as other land or other country speciation. And then the opposite of that, or the flip side of that, would be sympatric speciation, from the Greek syn, together. So same country or same land. So with allopatric speciation you have a geographic separation of the two groups, and with sympatric speciation you do not have a geographic separation, but there are other factors that cause the divergence to occur. So we're going to look at those two. We'll start with allopatric and then we'll move on to the sympatric. Your textbook always does a good job showing various illustrations, and they do the same thing here. So let's imagine we have a pond. You have a pond of individuals, a population of fish here, and then there are two possibilities for the way that a different species can arise, or that the population can diverge. On the left we have allopatric speciation, which is perhaps the most common way that this occurs, where you have a physical separation of the two groups. Let's say the water level drops, and so then you have basically two small ponds, and so at one time it was one large population that mixed alleles between them. You had gene flow between all of these various individuals. And then when you have allopatric speciation, a population forms a new species because they are geographically isolated from the parent population. This is perhaps the most common. We'll look at that in just a second. The other side of this is sympatric speciation. This would be where there is no geographic separation. There is no river or mountain range or something that's keeping the two populations apart, but still you have other factors we'll take a look at that cause a subset of the population to form a new species, even though there is no geographic separation, and that would be sympatric. So allopatric is like other country or different country. Sympatric means same land or same country. First up is allopatric speciation. Allopatric speciation is when gene flow is interrupted or reduced when a population becomes divided by geography. So that could be a mountain range or a stream or a river, or some change in the geography that for some reason isolates two groups of what once was the same population. A good example is earlier in the slideshow we saw the flightless cormorant of the Galapagos Islands. It is very similar to the cormorants that you find on the mainland that can fly and have some other different characteristics. So it likely originated from a flying species on the mainland, became isolated there at the Galapagos, and slowly diverged and changed until now it is considered its own species. The process of allopatric speciation kind of depends on what species we're talking about. If it's a small species or a species that doesn't have the ability to move quite a bit, it doesn't take much to separate them. For example, a canyon like this that you see here could easily create a barrier between populations of small rodents like chipmunks, but that same canyon may not be enough to separate a population of flying birds or coyotes that can travel, or plant pollen that can blow in the wind. So the definition of the barrier kind of depends on the creature that you're talking about. Once those populations become separated, they may begin to evolve independently through the same factors we discussed in the last chapter: mutation, natural selection, genetic drift, and then eventually you get to a point where there is reproductive isolation. Eventually you have enough changes that occur genetically, you have genetic divergence, to where they then, even if they were brought back together, those two populations have the various barriers we discussed that would keep them from breeding together. In your textbook they give the example of a particular small little fish called a mosquito fish in the Bahamas, that they are basically several different breeding populations, several isolated populations in different little ponds, and over time they have changed because the environments of the ponds differ slightly. Some of them have some other predatory larger fish that eat these little mosquito fish, and some of them are missing those predators. In the high predation ponds, selection has favored a particular body shape that enables the fish to move very quickly and escape predators. In the ponds that don't have those predators, evolution has favored, or natural selection has favored, a body shape that is a little bit different, that is actually a little bit better for swimming for long periods of time, but not for these great bursts of speed. The female mosquito fish in these various populations prefer to mate with the males whose body type matches theirs, whose body type is similar to theirs. So even if you were to put these two ponds together, you'll find that the females of one kind prefer to mate with the males of their same kind, and vice versa. So even if you were to bring these two populations together again, you wouldn't get the thorough mixing, the thorough gene flow, as if they were a single species. You can find a number of examples out there in the natural world for this allopatric speciation, and your book gives a couple of great examples. One has to do with some snapping shrimp that live down around Panama, near the Isthmus of Panama. At one time, this thin section of land, I'll show you a map, didn't exist. The water was high enough that you would have free flow of organisms from one side to the other of the isthmus, but now there is basically a land bridge that blocks that from happening. So there are about 15 pairs of what you might call sibling species of snapping shrimp that live around this Isthmus of Panama. And so what that means is you have about 15 species on one side, 15 species on the other side, and they are related to each other. So you may have like one species on the left of the isthmus that is most closely related to a species on the right, and you have that basically in 15 variations. So you have shrimp that on one side of the isthmus are actually more closely related to a shrimp that lives on the other side, more closely related to that one on the other side than to the others that live on the same side that it lives. And so this is a good example of allopatric speciation. In this area, as the land rose or the water level fell, you slowly begin to have this dividing line, and you had basically what was 15 large populations get split into two sections of 15 populations, and then over time those diverged, and now they are separate species. And as a matter of fact, this, through genetic testing, they kind of match up the time frame for how this speciation occurred, and it closely matches this time frame of when the Isthmus of Panama formed geologically, about at the same time period. And it even shows that the species that lived in the deepest waters diverged first, which means, of course, as the water level fell or the land level rose, I'm not sure what occurred there, that the ones that live in the deeper areas get separated first, and then the medium water areas get separated next, and then those that live in the shallow water get separated last. And that's exactly what the genetic testing shows as far as the divergence of the species as well. Take a look at a picture here to kind of understand. We're talking about way down here south of the United States, between the United States and South America, here in Central America, this little strip of land here, the Isthmus of Panama. At one time this was underwater, and you had the free flow of genetic material back and forth between all of these areas, but now they are separated from one another. And so you might have a species on this side that is most genetically related to now a different species over here, so they're called sibling species, and there's actually 15 different examples of that. So these were obviously at one time groups that were then separated through this geographic change, this geologic change right here, and then you had allopatric speciation occur. Other key pieces of evidence for allopatric speciation include the fact that areas or parts of the world, regions of the world that have a lot of geographic barriers, like mountainous areas and lots of streams or rivers or lots of different features that would easily separate populations, those areas tend to have more species than do regions with few barriers. So if you have a continent that is fairly flat and open and has fewer geographic barriers, you wouldn't expect to find as many species. And then if you find a place like, say, they use the example of Hawaii, not a continent-sized area, but just a smaller area, it has a lot of different geographically isolated parts to the islands, and you have a much wider variety of species there because you have all these tiny little separated areas that keep these groups apart. Reproductive isolation between populations generally increases as the distance between them increases as well. In the book they give the example of the scientists looking at a particular type of salamander, and they brought them into the laboratory to see if they could breed together, and the groups of salamanders that lived close to one another could successfully breed. But the further that they pulled species from, or the further that they pulled a salamander from a particular population, the ones that were further apart, they had greater genetic differences. They had greater speciation occurring between them. So the further apart the groups are, the less likely they are to be able to interbreed together. Now we know that's not always the case. Like we already mentioned, humans are spread throughout the entire world, of course, and they can still interbreed. But in many cases, especially for smaller animals, geographic separation is eventually what's going to lead to genetic separation as well. Here in your book is the chart that talks about those salamanders, and they were just showing how the degree of reproductive isolation goes up as the geographic distance between those two populations increases. One thing to keep in mind is that these barriers to reproduction are intrinsic. They are part of the genetic makeup or the biological makeup of the creature. So geographic separation itself is not a biological barrier. Humans are a good example. We have humans that are geographically separated around the entire globe, but that is not a biological barrier. And because human transportation and movement around the globe is so easy, relatively speaking, there's still a lot of gene flow, and so that hasn't developed into a biological barrier. But for many creatures who can't hop on a plane, geographic isolationism eventually does result in biological barriers that will then prevent interbreeding between those populations, even if you bring them back together. So for most creatures, if you geographically separate them for a long enough period of time, they will develop various biological barriers and genetic differences that even when you bring them back together, they can't interbreed. Your textbook uses a good example of a pretty well-known fruit fly experiment where some of the flies were raised on a starch medium food material and some of them were raised on a maltose medium food material. That was the only difference between the populations. They were taken initially from the same population, and then a number of generations were raised feeding on one material or feeding on another, and then when they were brought back together, you would find that they had a change. They had some differences between them, and the female flies preferred to mate with the male flies of their same kind. And so you began that speciation process just by separating the group and having them feed on different materials. The second way that speciation can occur is a little less common. It's called sympatric or same country speciation. In sympatric speciation, speciation takes place even though there's not a geographic separation. So this is speciation even though we're talking about a geographically overlapping population. So the question would be, of course, how can this happen? If you have nothing that separates the two populations and they can interbreed and there's gene flow, what would eventually keep them or make them into separate populations? There are a couple of things that can keep this from happening or that can begin to disrupt the gene flow between these populations. It is less common than allopatric speciation, but there's three factors we're going to look at called polyploidy, habitat differentiation, and sexual selection. And these three can begin to diverge the two populations even though they have overlapping geography. The first factor here we're going to look at is called polyploidy. Polyploidy is really common, or a lot more common, in plants. As a matter of fact, some of the most important agricultural crops that we use are polyploid plants. Not so common in animals really, but we still want to look at that because plants are an important part of the environment, of course, an important part of biology. So you will see this on occasion in other creatures, but plants is primarily what we're talking about. Polyploidy is the presence of extra sets of chromosomes. So if you think about poly meaning many, extra sets of chromosomes due to accidents during cell division. So for example, you might have a lack of chromosome separation during anaphase when the chromosomes are supposed to pull apart and then go back to their separate poles of the cell. You may have a lack of separation there and you can end up with extra sets of chromosomes. Again, much more common in plants than animals. As a matter of fact, botanists estimate that 80% of the plant species that are alive today either are polyploidy themselves or they have ancestors that formed via polyploid speciation. So we have a couple of terms here that we might want to know. An autopolyploid is an individual with more than 2 chromosome sets derived from one species. And an allopolyploid is a species with multiple sets of chromosomes derived from different species. You would like to think that they could come up with two words that didn't sound so alike, but they didn't do that. An autopolyploid is an individual with more than 2 chromosome sets derived from one species. An allopolyploid is a species with multiple sets of chromosomes derived from different species. So the only difference in between these two words is multiple sets of chromosomes from the same species, multiple sets of chromosomes from different species. As I mentioned, there are many important agricultural crops. Some of the biggest crops that we raise around the world, such as oats and cotton, potatoes and tobacco and wheat, are all polyploids. They all have more than 2 sets of chromosomes. They have multiple sets of certain chromosomes. And as a matter of fact, today there are plant geneticists that purposely breed polyploids using lab procedures to produce hybrids that have, the various desired qualities that we want, such as insect resistance or drought tolerance, things of that nature. Your textbook on page 496 gives a little diagram that shows an example of one mechanism for an allopolyploid speciation in plants. Remember, allopolyploid is where they have multiple sets of chromosomes from more than one species, from different species. So here you have species A and species B. Species A produces a normal gamete. Species B undergoes some sort of meiotic error in the copying process, and so it has a gamete that is doubled up in the number of chromosomes. It should have half that amount. And then when those two combine, you get a hybrid that has 7 chromosomes. Then again, you have species A is producing normal gametes, has all along, but now this hybrid has an unreduced gamete with 7 chromosomes. Again, another error in copying. And then that new species would actually be viable, a fertile hybrid, an allopolyploid, difficult to say, an allopolyploid, which has a 2n number of 10. So basically what we've created here is a new species, but that new species arised or was brought about through a reproductive error in one species and then combining with this second species. So now we have a third separate species that probably would not be able to interbreed with either one of the two parents or the two originators here. And again, this is very common, or a lot more common in plants, not so common in animals. Usually when you have this kind of meiotic error in animals, you're going to end up with a creature that cannot breed or reproduce at all, or a creature where the embryo undergoes some sort of a processing error where it isn't born, or it doesn't come to its full fruition before it's born. In animals, as opposed to polyploidy, you're more likely to have something happen like habitat differentiation. So in animals, if you want to see sympatric speciation, you might be looking at something like habitat differentiation, and that's where you have creatures develop their own little ecological niche, their own little place in the ecosystem. And a good example they use in the textbook is there's a particular type of fly called the North American maggot fly. It started out living on a type of tree called a hawthorn tree, which is native to North America. That's their original habitat. But as apple trees were introduced into their areas, some of them began to live on apple trees, and they began to use the apple tree as their habitat, as their niche. And so now there are 2 subspecies that are beginning to go through sympatric speciation due to their different food sources and habitats. So at the moment, these 2 groups of flies can still interbreed, but they are beginning to show differences, or beginning to show genetic differences and biological differences that eventually, given enough time, will keep them from breeding. And at the moment, they really don't interbreed much at all already, simply because of habitat differentiation. One of them tends to live on the apple trees, one of them tends to live on the hawthorn trees. They have a different food source, different habitats, and that begins to keep them apart. Lastly, again, more common in animals than, say, something like polyploidy, another type of sympatric speciation is sexual selection. Sexual selection can be a factor that begins to drive populations to differences. So sexual selection is for mates of a different color or a different variety. A good example is a type of fish called the cichlid fish. There's a lot of different examples, or a lot of different species of cichlid fish that live in Lake Victoria in Africa. And there's an experiment that the scientists have done using these fish to kind of show how sexual selection begins to diverge populations, and they used the different varieties of fish and the colors and lighting examples. So what happens was they took, say, 2 species of these cichlid fish, or 2 varieties of this cichlid fish, And you can see how under normal light the males have a different coloration and the females very much prefer a particular coloration. So this particular species, the females prefer the males that look like this, and in this species the females prefer the males that look like this, and so they don't usually interbreed with one another. But then what they did was, in the aquarium in the laboratory, they put on a monochromatic orange light which kind of washes out all the color. So now the fish see this, and the males of the two different species look very much alike. And what they found was, when they did that, the females weren't nearly as choosy. The females would tend to interbreed one with the other. So again, we're defining these as two different species, but they have the ability to interbreed. They're defined by their differences, not biological differences, but by some of their habitat and sexual selection differences. So when you washed out their colors and the females couldn't really tell them apart, they would interbreed more randomly. But when the females could tell the colors apart, they chose males that matched their particular species. So that's a good example of sympatric speciation through sexual selection. The groups are in the same area, in the same lake, but because of their sexual selection preferences, they tend to stay apart from each other. So let's do a quick review of those two different methods, allopatric and sympatric speciation. Allopatric, remember, means different country. Allopatric speciation is usually geographic isolation that restricts gene flow between the populations. Then, once they are separated, then you start to get reproductive isolation, biological isolation. That's because of natural selection, genetic drift, sexual selection in those isolated populations. And then once that occurs, once there are biological barriers between breeding, even if you were to restore those populations to one whole group, you would prevent interbreeding between them. In sympatric speciation, sympatric meaning same country, a reproductive barrier isolates a subset of the population without any sort of geographic separation. That's usually going to be the three things we talked about: polyploidy, which is normally in plants, differing natural selection pressures because of different habitat or diet change, or sexual selection. So we're going to pause right there, and those are some of the mechanisms or methods for speciation to occur. In a minute we're going to come back and look at hybrids, areas where species do overlap, and what happens when these species do breed and form hybrids. So we'll take a break. We'll be right back.